Hi and welcome to this video on the organisation topic where we will be looking at enzymes and how they work in the digestive system. To start off with, we need to talk about the food groups that get digested. One of those food groups is carbohydrates, and these get used in the body in respiration, so they are a big energy resource. And they are long chains of sugars, so when we break them down, we are releasing those individual sugars. One of your required practicals is to be aware of a variety of food tests for certain food groups. Starch is an example of a carbohydrate that you would find in any starchy food like rice, pasta, bread. And we could use a chemical called iodine, which is an orange-brown chemical, to test for starch. And if starch is there, iodine will turn to a blue-black colour. Sugars can be tested for using Benedict's reagent, which starts off blue. You would mix it with the sugar sample and you need to heat it to get it to work. But essentially, if there's a low level of sugar, you'll get a green colour. If there's a little bit more sugar, that green will turn into yellow. And if there's a very, very high level of sugar in the sample, you get this brick red colour. So kind of like traffic lights. Another food group is protein. And protein overall is used for growth and repair. But our enzymes are proteins, our antibodies are proteins, and quite a few of our hormones, like insulin, are protein-based. And proteins are long chains of amino acids. Once proteins have been broken down into amino acids, those small subunits can be absorbed into the bloodstream. We can test for the presence of a protein using a burette test. Again, we've got a blue chemical here, and in the presence of protein, we'll go kind of a pink or purple colour. The last food group I wanted to talk about are the fats and oils group, which are known more scientifically as lipids. And we need these fats and oils in our body because our cell membranes are made out of lipids. So are some of our hormones. Our nervous system relies on lipids too. And lipids actually also store quite a lot of energy. They get broken down or digested into glycerol, which is the large orange block that you can see here. And there are 3 fatty acid tails that are normally attached to that glycerol. And when lipids are broken down in the digestive system, they all get separated so that they are small enough to be absorbed into the bloodstream. To test for the presence of lipids, we could use a chemical called Sudan 3, and this chemical is red. And if you mix it with your food sample, if lipids are present, you will get a separation where the top layer will be a bright red colour. Alternatively, you could use ethanol, and if you mix ethanol with a lipid sample, it will go cloudy. So those are the food groups and how to test for them. And essentially, these major food groups are made up of long chains that are insoluble. They can get to our stomach, they can get through our small intestine, but they cannot get into our bloodstream. They're far too big. So digestion is all about breaking down these large insoluble molecules into smaller soluble molecules that can be absorbed into the blood. And this breakdown is sped up by enzymes. So let's look at how the digestive system works to break down these 3 food groups. The first point of call is the mouth. And here you have mechanical digestion, where the food is physically broken down by the teeth, but we're more concerned about the chemical digestion involving enzymes. So the enzyme involved here is released by the salivary glands to act in the mouth, and it breaks down carbohydrates into sugars using a class of enzyme called carbohydrase. And this particular carbohydrase enzyme is one that you may have used in class, and it's called amylase. From here, food then moves down the esophagus or food pipe and down into this organ here, which is the stomach. Here a different food group gets worked on, and that food group is the protein group. And proteins get broken down into amino acids. And the class of enzymes that break down proteins are called protease enzymes. And the particular protease enzyme that works in the stomach is called pepsin. So essentially the stomach is lined with glandular tissue, and this glandular tissue releases chemicals such as the protease enzyme, but it also releases hydrochloric acid. And this hydrochloric acid is there not to break down food, it doesn't do that, but its primary reason for being there is to kill the germs that could be on contaminated food and drink. And this hydrochloric acid creates the optimal conditions for pepsin to work, and that is pH, around about pH 2. Pepsin works very, very well at that pH. From the stomach, the food now has been partially digested, so starch has been partially digested, as has some protein. The food now gets squeezed from the stomach into the small intestine, which is this area here. And this part of the digestive system is all about absorbing the smaller, broken down, soluble components of the foods that have been consumed into the bloodstream. So the small intestine is the business. A lot is going on in the small intestine. There will be enzymes belonging to the carbohydrase group that will be acting on carbohydrates still in the small intestine. There will be enzymes belonging to the protease group as well, working on other proteins that have gotten through to this stage. But the final food group, fats or lipids, get broken down by a different enzyme here. So lipids are broken down by lipase enzymes into glycerol and fatty acids. The problem with fat digestion is fats don't mix very well with water. In fact, they don't mix at all. And the enzymes are water soluble, so this makes it very difficult for the lipase enzymes to gain access to a lot of the lipids because lipids clump together. So there is an extra chemical that helps with fat digestion, and that chemical is called bile, and it is made in the liver. This bile doesn't stay in the liver, it gets transported to this green organ here, which is called the gallbladder, which stores the bile. And when needed, that bile gets sent through this tube here, which is called the bile duct, which you can see just going behind the stomach here, into the small intestine. And how that works we'll explain in a moment. But there's one other organ I need to just mention briefly. It's very, very important in digestion. And that's this leaf-shaped organ here, which is called the pancreas. And the pancreas makes all three groups of enzymes. So it makes carbohydrases, proteases, and lipases. They don't actually work in the pancreas, they act and work on the small intestine. But majority of the enzymes involved in digestion are made in the pancreas, so it is a very important organ. Similarly, the small intestine does all three as well. So let me just explain briefly how bile is involved in lipid digestion. We've zoomed in here on a section of the small intestine, and flowing through the small intestine you can see a clump of lipids or a clump of fat, and each individual lipid is made up of one glycerol unit and 3 fatty acid tails. Bile that is made in the liver and stored in the gallbladder is sent into the small intestine via the bile duct, and it helps to break up this larger clump of fats or lipids into smaller clumps of lipids, and we call that emulsification. So it is effectively increasing the surface area of that larger clump of fat. So it is smaller little clumps of fat which give more access to the lipase enzymes that need to attack it and sever the bonds between the glycerol units and the fatty acid tails. So once lipase has acted on the entire group of lipids, you now have lots and lots and lots of small soluble pieces that could be easily absorbed into the bloodstream. The small intestine has a really, really large surface area because the inner surface of it is folded inwards many, many times to form these villi, as you can see here. If we zoom in on one single villus, you will see that there is a single layer of cells and there is a very rich blood supply, so that now that the lipase has acted on the lipids themselves, the glycerol now is small enough to fit through the semi-permeable membrane of the small intestine villi and also through the semi-permeable membrane of the blood vessel itself. Similarly, those fatty acid tails are small enough as well to fit through. And once these small subunits from the food are into the bloodstream, they get transported to the cells where they could be put to use. This glycerol and fatty acids could be reformed into a lipid molecule and could then be used to make a hormone or as part of the cell membrane of that cell. The amino acids that have been absorbed into the bloodstream, because that's going on here as well, proteins are also being digested by protease enzymes in the small intestine into amino acids. And those amino acids are now small enough to get through into the bloodstream. When they get transported to cells in the blood, they get made into new proteins that that cell needs. And the carbohydrates that will be going through the small intestine get broken down into sugars that go into the bloodstream to the cells and could be used as an energy resource for those cells. One other point to mention about bile is that not only does it emulsify these fats, giving them a larger surface area for lipase to work on, but the food that's going into the small intestine from the stomach has been mixed with a very strong acid. The enzymes that you find from the pancreas and the small intestine that are working in the small intestine do not like acidic conditions. They prefer slightly alkaline conditions, and bile, being a naturally alkaline solution, facilitates that. And it kind of creates a pH of around about pH 8. So a quick summary of all of this. So you have got your 3 food groups. So we would refer to these now in the context of enzymes as the substrates. So carbohydrates are broken down by a class of enzymes called carbohydrase enzymes. An example would be amylase. The products of carbohydrate digestion are always sugars. So you could give an example of a sugar like glucose or maltose. And carbohydrase enzymes are made in the small intestine and the pancreas, but also in the salivary glands. Protease enzymes break down proteins into the product amino acids. The example that you need to be aware of is pepsin, which is found in the stomach, but there are other protease enzymes made in the small intestine and the pancreas. Lipase enzymes are only made in the small intestine and the pancreas. There's no additional other location in the digestive system where these are made, and these break down lipids, also known as fats, into fatty acids and glycerol. So hopefully this bite-sized revision video on enzymes in digestion has helped your understanding of it.